Welcome everybody to this H3D Foundation webinar. I'm Nicola Elliott-Wong. I'm the program coordinator at H3D Foundation, and it's my great pleasure and honor to host these sessions. Today, we're very blessed to have Karsten Menzel from MSD join us to talk about long-acting injectable drug design. And I'm going to start by reading his bio to kick us off. As a senior principal scientist at DMPK at MSD, Karsten Menzel provides decisive scientific and strategic leadership to drive drug discovery and development programs forward. His work spans diverse therapeutic areas, including infectious diseases, cardiometabolic disease, oncology, and neuroscience, with a proven track record of advancing compounds from discovery through first-in-human studies and ultimately to regulatory approval. His expertise is in characterizing small molecule and biologics ADME properties, predicting human efficacious doses, and assessing drug-to-drug interaction liabilities to shape robust clinical strategies and support successful global regulatory submissions. He earned his Ph.D. in organic chemistry from Philipps-University Marburg and conducted postdoctoral research at MIT. As part of the MSD Fellowship for Global Health, he was instrumental in building and expanding DMPK capabilities for tuberculosis drug discovery through the TBDA program at the Infectious Disease Research Institute. His current focus is on applying model-informed drug discovery to enhance research in resource-limited settings and integrating machine learning models to accelerate daily decision-making in drug discovery. Karsten, we're so happy to have you today. Thank you so much for carving out the time. I know you've been working with one of our staff at H3D and Chinda in a very focused way to kind of create this bespoke webinar for us. We're so thrilled to have you. To everybody, thank you for being here as always. Please populate the chat with your questions, and we look forward to a robust Q&A session at the end of the webinar. Over to you, Karsten.
Thank you so much, Nicola, for the nice introduction, and welcome everyone to this webinar. So as Nicola mentioned, so we are going to talk about today about long-acting injectables. So for me, even so, I love working on this topic because it's a little bit different as compared to oral drug discovery. I'm a little bit afraid of, so getting injectables, so with a needle injected in myself. I don't know how you feel about it, but for me, it's oftentimes like not the preferred way how to administer a drug. But I think the long-acting injectables have their utility and their place in delivering medicines across the world. So today I want to talk about long-acting injectables from the discovery and translational perspective. So the core idea I want you to keep in mind throughout this talk is that LAIs are not something that you fix at the end with formulations. It's actually a designed product that can succeed and fail based on early decisions that you're taking throughout the drug design. I will walk you through how clinical objectives, the medicinal chemistry effort, PK principles, formulation constraints, but as well development realities all connect. So the goal is not to provide a the goal is to provide a framework for thinking about how LAI feasibility can be assessed early and to minimize the risk for compounds that are not really suitable for an LAI and to risk them relatively early at each stage as a step of the discovery and development effort. So LAIs, as I pointed out initially, so they do have their place in delivering medicines, but they are a niche product overall. But there are a couple of products on the market that are really successful, and they have been primarily, so what I have collected here are basically a couple of therapeutic areas like psychiatric disorder or hormone therapies, and lately as well infectious diseases. And you see these kind of drugs are primarily around the treatment and prevention of HIV. So the drugs are on the market, and they are being administered with a duration of 1 month. They can exceed even 3 months or longer, 6 months, what you see here for 2 of those products. But those kind of variations in duration, they are reflect basically clinical objectives. So you want to have maybe maintenance therapy, you want to improve adherence, or you want basically to provide a prophylaxis options for certain indications. Now, there are as well active research going on in other, not in other therapeutic areas, but within a therapeutic area like tuberculosis and malaria, which might be actually close to your heart. What is going on there? So I collected a little bit of information on options or candidates that are currently in development. So none of those kind of products are actually on the market at the moment. But there is active research going on in the clinical and the preclinical space. So for tuberculosis, there are 2 drugs candidates currently in the preclinical, in the development space. So in phase 1, where long-acting formulations are being assessed in humans, with the idea of prevention of TB. The same is true as well for malaria. So there's 1 drug as well in the preclinical space, in the clinical space for testing a nanoparticle suspension of atopic for the prevention of. You see, for example, I put as well the duration on there. Most of them are trying to target a less frequent dosing, so 4 times a year. But if you could, for example, convert this into a 3 to 6 month duration or dosing frequency, that could be really an transformative application of this kind of medicines in the patient population that we are talking about. Now, when we start thinking about formulation, then there are a couple of options that you can select from. The workhorse, so to say, for LAIs are either solutions or suspensions. And most of the marketed LAI drugs are suspensions. So, and the reason I think is relatively obvious, so they oftentimes tend to have a low aqueous solubility, which results in a low dissolution and prolonged release of the drug from the depot site. And this is an important theme that will come up repeatedly. So the formulation choice is not something that is an independent decision that you can take. It's actually dictated by dose and the intrinsic property of a molecule. Now, alternative other formulation platforms are, for example, polymer-based systems like microparticles, in situ gels, or implants. The trade-off here is actually the dose. You saw on the previous slide that the total dose that you can administer are higher than for those kind of polymer-based systems. And those kind of systems, so the polymer-based systems are typically required very low total drug loads, and they have as well a specific solubility behavior for organic excipients due to the process how they are being manufactured. So while they can offer a longer duration, so what you see here, 1 month to a year, they narrow actually the range of molecules that you can select from. So and the driver for the solution is actually here the degradation of the polymer-based systems rather than in solution and suspensions, where the driver is the low solubility of the molecules. So another consideration that that is important from a practical perspective for the development of LAI is actually where you're going to inject the drug, because this might lead into limitations of the injection site volume. So on the left-hand side, you see intramuscular injections. On the right-hand side, subcutaneous injection sites. and they have different volume limits, how much per injection you can actually administer. So with IM, typically your volume that you can administer is higher as compared to subcutaneous dosing. So if you just think about how much drug you can administer in a certain volume, typically then your IM injection allows you to administer more drug load per volume administered as compared to the subcutaneous administration. And that contributes, for example, to a success of an LAI that you're selecting and for a candidate. Now, here on this slide, this is a principle that's really close to my heart. So the question oftentimes come up is, why do long-acting injectables last so long? When you think about typical oral dosing where the elimination half-life, maybe I'm turning on the pointer, where the elimination half-life in this area here dictates the dosing frequency. So how often do you need to give your drug? So whether it's BID dosing or once a day dosing, for example, or less frequent dosing, like once a week. This is being determined by the elimination half-life of your drug. This is different actually for long-acting injectables. Here, the pharmacokinetics are actually are considered flip-flop kinetics, where the absorption half-life or the absorption by itself dictates the how long the drug remains in the body. So that means that the absorption rate constant, Ka, controls how long the drug lasts in the body. This is a crucial, in my mind, that's a very crucial mindset shift as compared to your oral dosing. So long-acting LAIs are long-acting because the drug enters the system slowly, not because the body clears it slowly. Now, what information do we need to collect for assessing whether a candidate has the potential to deliver or can be delivered as an LAI? So for that, we need to align the BD target. So what kind of exposure do you need to achieve in the systemic circulation? The PK, so how fast it's going to be eliminated, because that dictates the concentration that you're achieving. And then, of course, what kind of dosing interval do you want to, or do you need? So from a clinical objective perspective, so is 1 month enough, or do you need actually to go with once a year or twice a year? So it's not just enough to hit your target concentration once. You need actually to maintain this efficacious concentration throughout the entire dosing interval. And because it's going to be an infrequent dosing of month, so it's so important actually that the compound is constantly delivered over on a daily basis. So in addition to the dosing duration, so what I want you to think about as well, because it's less frequent dosing as compared to once daily dosing, and so people are maybe not used to, so to think about your medication then on a daily basis. You need also to think about, for example, patients' physician office visits, so how often do they actually can come to the office? So visit cadence, for example, or in addition as well, forgiveness windows, so if someone is going to give to get a 6-month injection, can they actually, so is there a possibility to have a 2-week window where they can come back into the office? So plus minus 2 weeks, for example, might be an important factor to take in consideration, and that will contribute to your dose determination. Now, how do you translate those kind of information that you need to collect for an LAI in a screening funnel? Early on, we can use our in silico design for designing LAI compounds, We need typically, like for oral drugs as well, to assess the in vitro potency. That hopefully is an assay that translates into clinical efficacy. And then you want to assess as well the pharmacokinetic parameter of this kind of drug. These kind of two informations there, potency and PK, allows you actually to do an early LAI prediction. And I go into this in a couple of slides down the road. But this allows you an early triaging of compounds for LAI. If those kind of compounds actually look promising from an LAI perspective, you need to resynthesize the compound because the next steps are actually relatively resource intense. So you need to understand the physical chemical properties of your compound more thoroughly. So for example, your crystalline solubility. So you need to crystallize your molecule. You need to generate a melting point ideally. And then on top of it, you want to have formulation screens done for primarily actually for the long-acting injectable formulation. And this is not typically just one formulation, but multiple formulations that you're testing there from an in vitro perspective. And then you're going to test those kind of formulations in an in vivo model in order to see how the release profile looks like from the formulation, from the depot that is being administered in the preclinical model, and how that translates over into a concentration time profile. Now, once that actually looks promising and, so might be amenable as well for an LAI in human, this is where you're going to select a candidate for advanced studies, so like LAI studies in large animals. And from a medicinal chemistry perspective, you need then to resynthesize the compound again, so where you're generating significantly more material to do those kind of large animal studies, or you want to test it as well in an in vivo efficacy study. If this is a new compound and you still have translational concerns from your in vitro assay going into your in vivo model. But when all of those kind of results are checking, so that could be your clinical candidate in order to move into the clinic. So the goal here, so with this kind of screening funnel is to identify which of those compounds are worth advancing in more resource-intense studies. So you see, so from relatively low resources in the beginning, and then more resource requirements towards the end. So for only a few compounds to select from. And this is an iterative process that I would like to mention here as well. So you're probably spending a lot of time up here, and then less time here, and only really few candidates actually going to go into these kind of efficacy in large animal studies. Now, the question from a medicinal chemistry perspective is, what can you do in order to help designing long-acting injectables? So for your typical oral program, so solubility is supposed to be high, right? So this is your driver here. For long-acting injectables, actually, the low solubility is not a problem. It's actually a design feature, right? So you want to design low aqueous solubility compounds. So you have a couple of assessment points here, so with regard to your molecules should remain largely non-ionized at physiological pA, so that could mean, for example, reducing hydrogen bonding. So typically what you don't want to do, so you want actually to have some hydrogen bonds. You want to design high melting point compounds, for example, through rigid flat molecules, so that are densely packed in the crystal structure, so with strong intermolecular interactions. You could as well, for example, go in with high lipophilic compound, or so high lipophilicity, so that you have limited drug hydration or dissolution at the injection site. I want to caution you there a little bit because, so when you go in with higher lipophilicity drugs or compounds, then you need to balance that a little bit with the PD efficacy, so unbound drug, and as well DMPK properties, like that your compound might actually undergo relatively quick metabolism or elimination from the systemic circulation, or have a lot of non-specific binding. But nevertheless, so what there is a trend analysis being done, for example, between melting point and lipophilicity. And some of the drugs, so you see here, you can be successful actually with the long-acting injectables if you have a high melting point and be in a typical lipophilicity range from a normal drug area, so 1 to 4, 1 to 3. But you have as well some of the drugs that are on the market where you have a low melting point and long log P. So there's these kind of opportunities where you can go either way and develop a drug. And this is what I have tried to summarize here on the table, where you see the marketed drugs that I showed you on the first slide from a psychiatric disorder and hormone therapy, infectious disease perspective. All of those drugs that you see here have actually low solubility. Please ignore this one here for the time being, but so all of them actually have low aqueous solubility, and you see that they have high melting points, a relatively low log P, or they have a low melting point and a high log P. So this one here, for example, leuprorelin, this has a high aqueous solubility, and you see as well that this is a mismatch. So you have a low melting point and a low log P, so that relates actually into a high solubility drug. This compound here is highly potent, so therefore you can actually develop this as a formulation, as an LAI formulation, and you need some measures in order to not deplete the depot very quickly, so through, for example, polymer-based system. What can you do if your compound has all the characteristics for an LAI? So it's very potent, has relatively low clearance, so therefore the predicted long-acting injectable dose is supposed to be actually low. However, your physicochemical properties is not in the right space. So the solubility is too high, and therefore your concern is that the depot is depleting too fast. You can reduce the solubility through prodrug strategies. You most likely know that prodrugs have been used in order to increase the solubility. Yes, that is one area you can go to. But here for LAIs, you can do the opposite way, where you can introduce pro-moieties to your drug that hopefully contains hydrophilic groups that can be attached, so like you see here in some of the examples from paliperidone or aripiprazole, that you're attaching a carboxylic acid onto the molecule in order to reduce the overall lipophilicity of the compound, and as well increase the crystallinity and lowering the solubility. So this is a common strategy for LAIs and have been successfully applied in the LAI space. Now, the other opportunity is, which is definitely more difficult to achieve, is generating a salt. And it's not your typical TFA and HCl salt. Your counterion is typically a little bit larger. You need to make sure that you have that your salt has a high degree of crystallinity, low hygroscopicity, so typically something that you don't achieve with an HCl or a TFA salt. And of course, it needs to have a stability in the solid state that is amenable, for example, under accelerated conditions. So there is an example, olanzapine, that has been converted over into a salt in order to manipulate the solubility of olanzapine, and is successfully marketed as a drug. Now, the other possibility is to modulate the release rate of the compound from the depot. Is not only the aqueous solubility, but you can modulate it as well with a particle size. This play with particle sizes might end up or give you maybe a 50% or twofold variation in your release rate. And you can play, for example, with large and small particle size distributions of your suspensions. So this is particularly important for suspensions, of course. So if you control your particle size in a small way, so then you can, for example, increase the dissolution, or if you have large particle sizes, you might actually slow down your release rate from the depot. One caveat there is the larger your particles are going to be, the higher likelihood is going to be that you have injection site tolerability issues. So you might actually need to use larger needle gauges in order to deliver your formulation, which could cause pain for the patient. And so then the adherence or taking the drug might not be there. Another observation that was published in the literature is there was some observations about a medium-sized particle size, and that is reported anywhere between 1 to 10 microns. That could actually have an underside immune response, and I'm going to go into this a little bit later on. So what I want to take away from here is basically that you need to do some experimental evaluation of particle sizes and LAI formulations to understand basically what the release rate might look like from your depot. So it's not something that you can predict just from models. Now, how do you? So I talked about so that you need potency and PK early on to assess whether your compound or your candidate can be an LAI compound. And what you need what you can use in order to do this kind of assessment is a simple approximation by using the predicted human clearance based on your animal model PK that you have, times the target that you need to achieve, and then as well the duration that you want to go for. So let's say you want to go with a 3-month injectable, then it would be your 90 days. If you factor in, for example, forgiveness window, let's say you want to have 100-day coverage of your dose. So you're taking these 3 parameters, and that gives you the minimal long-acting injectable dose that you need to go after. And I think it's a common understanding, so if you have a low clearance, that means you have a smaller dose. If you have high potent compounds, so with low circulating or targeted plasma concentrations, you have going to have a low dose. And the longer you go, the larger your dose is going to be. And that is as well something that you want to keep in the back of your mind is the longer your duration is going to be, the more constraints you're going to have for your LAI compound. And this is maybe illustrated here in the middle part in this plot. So if you have compounds with relatively low clearance and high potency, then you're ending up in this green range here where you have a lot of freedom to operate with regard to the duration that you can formulate. And there are a couple of drugs in here that are amenable to this. If your drugs have properties from a human clearance perspective or from a potency perspective that might be outside of the ideal clearance and potency range, you still will be able actually to develop this as an LAI. However, your duration might not be as long as you might actually want to have it. You might not be able to, so for those 2 compounds here, to develop then as a 6-month or once a year long-acting injectable. And if, of course, the clearance is too high, the potency is too high, outside of that range, you will not be able to develop that as even a 1-month LAI drug. Now with this, in order to calculate the long-acting injectable dose, this is borrowed from an infusion equation. So you have clearance, you have your target, you have the duration. That basically is a normal IV infusion equation that you can take. But and this is your initial clinical LAI dose. The problem here, or not the problem is, so this is a minimal dose that you need in order for the drug to be released from the depot. And, but that is not reflective of any kind of bioavailability that you might need to factor in as well from the LAI. And that has been done. There has been done some retrospective analysis of marketed drugs, so how they translate the dose from a preclinical model to humans. And this kind of infusion equation, provides you a 2 to 5-fold under prediction of the actual clinical dose. So if you're going in and you're generating in your screening funnel potency, that's going to be your plasma target concentration plus your predicted human clearance, and multiply this by the duration, this is your minimal LAI dose. And if you're applying now a 2 to 5-fold factor on top of it, this gives you the range that you might gonna end up, that you're looking at for the human LAI dose eventually. So it's a useful equation to very early on, so where you only need 2 data points basically to assess whether you have an LAI candidate available or not. And so I think this is a very useful, powerful tool to use. Now, what you want to avoid is a little bit to have extremes. One thing is, if your solubility is too low, your drug might release actually very slowly, and you're going to see something where your drug concentration is never going to hit your C trough target that you need to achieve. It could be as well that maybe your drug is releasing too fast, and therefore the duration is not there that you want to hit. So instead of, let's say, a 3-month goal, you want to be able to actually deliver a 1 month with the drug load that you have. And so there is going to be a sweet spot eventually, so where you have the right amount of solubility, the sufficient potency of your compound to maintain the level for the intended duration of your LAI. The assessment of LAI formulations in the preclinical settings can be done in multiple models, as I pointed out in the screening funnel. Your workhorse here is primarily going to be the rat. They have a little bit of limitations, right? So because of the size of the animal model, you have limits with regard to the volume of your formulation that you can administer. What has been published in the literature is typically like top dose of, let's say, 300 microliters. So therefore, your dose is going to be limited depending on how much drug load you can deliver in this kind of volume of 300 microliters. The advantage here is rat are really accessible resources, and you can use them, on a regular basis. The assessment of your formulation, LAI formulation, in larger animals like in a rabbit, dog, and non-human primates, they are, so the resources are less available, but at the same time, you have the opportunity actually to test the LAI dose that you would like to give actually in humans, because you can administer basically the same volume of your LAI formulation as in humans. Now, one thing that I highlighted in the beginning was that with especially IM LAI suspension, you can observe, for example, or you want to look out for injection site reactions. And one of them actually has been described for paliperidone development formulations, where they observed when the LAI formulation was administered IM, that around the depot site and in the depot site, they saw macrophage infiltration and angiogenesis, meaning that the drug had a higher release rate as compared to what they expected from the solubility of the compound itself. So this illustrates as well that some of the, even so you can plan for the candidate to be developed as an LAI, you need to do many experimentation in order to assess whether the LAI formulation really performs as you intended actually to have the performance done. So from a screening funnel perspective, so you're gonna test your LAI formulation in the rat. We talked about the limitations in the rat. Once those kind of release profiles and duration looks good, looks accessible or acceptable for your profile, then you move into a large animal study in order to confirm that the formulation that you develop for your LAI is actually performing as good as in your rat. There's active research going on that you can use actually the rat release profile from the depot to translate over into human. There is some success in this kind of in this kind of translational aspect. The large animal at the moment, from the literature perspective, is still the way to go with translation of the release profile to humans. However, my expectation is that rat is going to be in the future a suitable animal model to translate this kind of information all the way to humans, and you don't necessarily need actually large animals any longer, or only for a confirmation perspective. On the right-hand side, for example, there was a recent publication last year about comparing the absorption rate constants determined from an LAI formulation in the rat, translating or comparing that to the absorption rate constant in humans. And either allometric scaling or linear regression actually gave you a very similar result, and they allowed you to translate. So if you determine the absorption rate constant in rat, you can actually predict what the rate constant is going to be in human, and you have a better understanding of is your LAI formulation performing as you need to have them performed for the duration of the study. And I would like to close out the presentation with just one slide about clinical development for LAIs. The LAI development in the clinic is more tedious as compared to an oral paradigm. On the right-hand side, in this first box, you see, for example, for cabotegravir and lenacapavir, which are LAI compounds for the prevention and treatment of HIV infections. The number of phase 1 studies to assess LAI formulations are so 3 to 4 times higher as compared to your normal oral drug development. So you need to invest more into phase 1 studies for the assessment of LAI as compared to a normal oral drug program. And I think that's what we need to keep in the back of our mind, that the resources in the early clinical space is actually more than for an oral drug development. What you need to take in consideration as well, because those kind of LAI studies last longer, you need to have as well, from a regulatory perspective, most likely the requirement for chronic dosing data in animals in order to support your 1 to 3 or 6 month LAI dosing. And this is typically something that you need to think about when you go into an LAI development early on, that you might need these kind of data in the preclinical species in order to maintain the safety and tolerability in humans. So when you think about what kind of studies are you going to do in a phase 1, so you want to have, if this is a new drug that has never seen actually a human being, you want to have safety and tolerability assessed, ideally with a short-acting drug that could be, for example, oral, if the drug has not such a low oral solubility that it cannot be administered as an oral drug, then you need to find a different way how to assess safety and tolerability in humans first. In an ideal situation, you want to have an IV PK study done in order to get the clearance, because the clearance can be a good surrogate for testing out whether your compound, so your translation from preclinical clearance to human clearance is actually what you predicted and still maintains an LAI dose that can be developed. You might want to have the opportunity to test proof of biology of your compound if it has never seen in human, just because the next steps for LAI development, formulation development is so significant that you want to make sure that your PK and your efficacy is in the ballpark where you need it to be. And once you have all those kind of information that typically are as well being done, except of the IBPK study for an oral drug, you're going to go into short-acting injectables and or long-acting injectables, optimizations of your formulation, and develop your drug for the patient population and the clinical objectives, so how long you want to administer the drug. And with that, I like to wrap up this kind of presentation. So the LAIs, hopefully you have seen that LAIs can be designed, that they are designed systems or products that, especially if you originate them in a discovery setting, that potency, solubility, in part as well clearance are non-negotiable, so that chemistry can contribute to the design of the molecules, that formulations are critical for the development of a suitable LAI dose, and that all of it, chemistry, formulation, and as well the PK, needs to be integrated from the start, and that you have the opportunity actually to do this very early on for this kind of assessment. And what I want to highlight as well is that iteration here is very important, right? So you need to iterate experimentally the LAI formulations, your compound design, so that you can identify candidates that are that can be successful LAI candidates. So if you embrace this kind of mindset of iterations early on, we can improve basically the odds of our success. And with that, I thank you for listening in and looking forward to any kind of questions that you might have.
Karsten, thank you so much. I hope I'm coming through okay. I just got bounced out of my own webinar by Teams, but I'm seeing the applause coming through. And there's a question in the chat. Farhat Ali from AIBST, would you like to unmute yourself and ask your question?
Okay, thank you. Thank you for the nice presentation, Prof. I just wanted to know during the solid state characterization of amorphous NCE, so how glass transition temperature directly influence the flip-flop pharmacokinetics in LAIs?
That is an excellent question, and unfortunately, I don't have a good answer for you. So whether that actually has an impact overall to your, so whether that can translate over into a measurable difference in your release rate from the depot. Unfortunately, I don't have a good answer for you.
Okay.
Sorry, I was on mute. Gosh, what a day. Okay, Stanley, I've got a question from Stanley, and I'm going to actually suggest that everybody raises their hands as we have a few lined up. So Colin, go ahead.
Thank you very much for the presentation. I noted that you were focusing a lot on the solubility as a key driver for the LAI, and there was less discussion on the elimination, the metabolic elimination. How important is it in the general design, or are you just banking on the fact that the low solubility and the low Ka absorption constant are going to take over and minimize the importance of K elimination?
Yeah. No, that's an excellent question. The clearance is actually as well an important parameter, as I pointed out. When you think about the infusion equation that I highlighted, the clearance is actually a critical parameter property to assess whether a compound has legs for the development of an LAI. The clearance of a drug will dictate, in combination with the absorption rate constant, how high your concentration, how high the concentration is going to be that you can achieve in the drug. So, in your systemic circulation. So clearance is an important parameter. However, what is important as well is, for example, the elimination half-life of the compound, but it's not as critical, so it's not determining basically how long the LAI duration will last. This is really driven through the absorption half-life, so how slow the drug is being released from the depot, that will give you the duration of the drug. The concentration that can be achieved is a combination out of how fast the drug is going to be released from the depot, plus how quickly is the drug being eliminated from the systemic circulation. So the lower your clearance is going to be, the higher your drug, depending on if you maintain basically the same absorption rate constant, so how much goes in on a daily basis, you can manipulate basically the drug. So you want to have a drug that is relatively slowly eliminated, that is correct, and you want to have some from an LAI perspective, you want to have as well a drug that is slowly released from the depot, but the combination out of both will dictate basically whether you achieve the target plasma concentration that you need to achieve. So it is important to pay attention to clearance, and this is why in your screening funnel you have relatively early on your assessment of the PK in the preclinical model, because that is one key parameter to look for the assessment of a candidate as LAI.
That answers your question, Colin, and I'm going to hand over to Candice from H3D.
Hi, Karsten. Thanks for the fantastic talk. There were so many great tips there. I'm interested to get a bit more information on the oral safety and tolerability studies. So this is something that we've been discussing internally. Could you maybe highlight what are those different ways that you could do this if you don't have a compound that has a good oral fraction absorbed? And just on addition to that, if you have a compound that has a really low solubility and permeability, and perhaps not opportunities to make a prodrug, for example, so that it can go into an oral safety study, would you flag that as a risk already early on, as a very difficult way forward to develop an LAI?
Yeah, that's an excellent question as well. Yeah, so if you have a compound that cannot really be developed as an oral delivery form, because the oral bioavailability is just so low that you will not achieve high enough concentrations. The option that you might gonna have is, and you need to explore this with different formulations, for example, to do your first in human studies with a new drug candidate using a short-acting injectable, for example. What I pointed out here in this development paradigm is your short-acting injectable, comes in here, where you already have potential, ideally, so from an oral administration, some safety tolerability, but maybe if this is not an option here with an oral drug delivery, you might need to invest in formulation work that helps you to deliver the drug in a short-acting injectable formulation. Meaning that instead of, so maybe if you're going to go after 3 months, so maybe this only lasts at high enough concentration that you can test actually safety tolerability. That maybe lasts 1, 2, 3 weeks the most. And, so then the concentration is still coming out of the, so the depot is still releasing the compound, but at very low concentrations, which are not then considered to be so problematic, but you have the opportunity basically to test out safety and tolerability. So that is one option that you can apply if the compound from an oral administration perspective is not applicable, or whether, for example, salt or prodrug efforts are not really amenable to for your drug to come up with an oral drug. But even, so for example, if you're going to go with a prodrug, then you have a new chemical entity that you're testing out here. And yes, you will test, of course, as well the safety tolerability of your parent drug, so which is the API or NCE, but this definitely makes it much more complex as compared to if you can develop an oral formulation of your drug, for sure.
Okay, thank you. That's helpful. And do you have any examples where they've looked at slow release IV infusion for safety? I was wondering if you.
I have not seen that actually in the literature for the marketed drugs. They typically went in, so what you probably appreciate is in the psychiatric disorder and as well hormone therapy, most of those drugs actually were developed with an oral formulation in mind, and then retrospectively fitted them into an LAI just because of the patient population and adherence and compliance perspective. Now the infectious disease area, they came in actually differently, so they designed new compounds from in the discovery space with the idea to develop them as an LAI, and they came in actually with an oral formulation. So they didn't have an IV infusion selected for those kind of compounds.
Okay.
So I'm not aware of an example there.
Okay. Thanks so much.
Yep. I mean, there are some infusion studies done, IV infusion studies done for longer acting compounds, but it's a different principle. So for example, antibiotics, some of them have a very long half-life. So the driver here for the long acting part is actually the half-life of the compound, so it's just circulating, it's the systemic circulation, it's not coming out of it or being released from the depot. So they have been using IV infusion, but this is a different scenario, or an example that is not applicable to a long-acting injectable that is being derived from a depot.
Okay, great. Thank you. Moving over to Matthew, also from H3D.
Thank you, Karsten. This was a really good webinar, and I think we've learned a lot that we can use. So what I wanted to ask is beyond solubility measurement, because what we are thinking about here is release rates and how they'll compare to the elimination rate constant. So what I wanted to ask is whether in vitro dissolution rate is measurable well enough that it can be predictive of in vivo release rate.
Yeah, this is an excellent question. There are in vitro experiments that you can do to measure the dissolution rate of a compound from an LAI formulation. And people claim as well that you can use that in a rank ordering fashion to select formulations to be tested in vivo. My answer to this is I haven't had success on the programs that I'm supporting from a long-acting injectable perspective to be translatable, so from an in vitro release profile, that this translates even from a rank ordering perspective over into an in vivo release rate perspective. So my personal experience is that I don't see this kind of trend, but researchers in the literature have commented on that you can use the in vitro experimentation release rate determination there to rank order compounds for LAI formulation. So I definitely would use it and experiment with it, so whether you can establish this in vitro to in vivo correlation or rank ordering for that reason.
Thank you so much.
Dianda, over to you, also from H3D.
Thank you for a very informative talk. I already asked my question in the chat. I'll just read it out loud. So what results would you be looking for in an oral PK study to decide if your compound as a potential LAI?
Yeah, so the oral PK study is meant into the earlier question, so in support of that, so you for the translation basically or preparation of a clinical development paradigm. You by testing out oral formulations in preclinical models, you will get an idea whether you will have the opportunity to develop the drug as a short-acting oral formulation to test safety and tolerability. This is really the only idea why you want to take a look at this. There's no other particular reason actually why you want to test. So you want to have basically the oral bioavailability tested in preclinical species with an oral formulation in order to see whether you're going to see 2% of an oral bioavailability or whether you can actually get higher exposures.
Let's say if you have an oral bioavailability of 50, would that compound still be good to be developed as an LAI?
Can you please repeat the question?
I'm saying in terms of, let's say you have a bioavailability of 50, would that be still good for your compound to be developed as an LAI? Would you suggest for someone to develop it
Right.
as an LAI?
Yeah. Because the oral formulation can actually, so you can have maybe excipients in your oral formulation that boosts maybe your oral bioavailability. So you could have, for example, as well, the administration of a spray-dried formulation. This is an amorphous drug of your, or an amorphous form of your drug, which typically results in a higher solubility as compared to a crystalline form. So you could, for example, end up with an oral bioavailability of 50%, but that does not mean that your release rate from the depot of a crystalline form of your compound actually has a low release rate. So there's definitely a possibility that you have a high or relatively high oral bioavailability because your oral formulation is different as compared to your LAI formulation.
Thank you so much.
And maybe one answer, so why the oral formulation might be useful as well as if you think about the development in the clinic, you always have as well a preclinical safety profile program that you need to work on. And so doing your safety profile in preclinical species with an oral formulation is significantly easier as compared to doing all of this with an injectable. And so an oral formulation will help you not only for testing safety tolerability in the clinic, but as well to achieve high enough exposures in your preclinical safety program.
That makes sense. Thank you so much.
We're going to end off with some questions from Stanley from AIBST. I know he's been waiting a while. Thank you, Stanley.
Okay. Hello, can you hear me?
Yeah.
Yeah, okay. So quick questions. Is there any role of the variation in drug metabolizing enzymes within the skin in terms of the efficacy of the LAIs? And the other one, maybe you may have answered it one way or the other. And just like the reverse, how feasible is it to convert an oral drug into an injectable? And are there any advantages in that route?
So your first question was about metabolizing enzymes, whether they have. I'm sorry?
Yeah, the variations in the drug metabolizing enzymes.
Variations, or you mean going from preclinical species to humans?
I mean, having different forms of the enzymes within the skin that may metabolize the drug faster or slower.
Yeah. No, so from a mechanistic perspective, how the drug is going to be eliminated is, so I would not pay too much attention to this in the beginning. Of course, you need to describe it eventually, how your compound is being eliminated. So you need to have some understanding, but using it as a driver for developing an LAI candidate, I would be cautious with this, and I would focus on the totality of the elimination pathways by just measuring the clearance of the drug. If you want to use it from a design perspective, so that you want to understand, for example, how to design in a medicinal chemistry perspective, maybe, or influence the clearance, and your compound is being eliminated through drug metabolizing enzymes, there might be a value eventually to know what kind of enzymes are involved in the elimination of a drug. And you might want to take a look at, so soft spots and, so where the compound has been metabolized in order to reduce those kind of side of metabolism. But overall, so just looking at the clearance of the drug is probably the way to go, and much faster as compared to anything else. Your second question was?
Is there any advantage, or is it feasible to convert an oral drug into an injectable? I mean,
right.
Would it make sense? Yeah.
Yeah, so oral drugs can be converted into LAIs, because that was the typical way previously, especially in the psychiatric disorder drugs, to move them over from an oral drug into an LAI, or for example as well the current HIV medications, lenacapavir, cabotegravir, rilpivirine. All of them are actually available as oral drugs, and they are as well developed as an LAI, so they are amenable for that. So you can definitely do this in that way as well. So if you have an oral drug, if the physicochemical properties are all amenable with an LAI, that is definitely the way to go. Or if, for example, from a strategy perspective, your compound is orally available, and you have, for example, some anchors in your molecule that could allow you to convert it over into a prodrug in order to manipulate your physicochemical properties that makes it more amenable for an LAI, then I think this is maybe the way to go. So definitely a yes.
Thank you.
You're welcome.
Okay, I think that brings us to the end of our time with Karsten. So it remains for me to say a huge thank you to our attendees to show up week after week. Thank you so much for your questions at the end of the session in particular. And a huge thank you to Karsten Menzel for giving us your knowledge, your passion, and your expertise so generously today. It's definitely landed amongst our, in particular, AIBST and our H3D members. So thank you so much for doing this for us, and we hope you'll be back in the future.
Thank you so much, Nikolai, and thank you so much for the excellent questions. Appreciate it, and thank you so much for the invitation. I really enjoyed it. Thanks.